Hi, hello, this is Dr. Swapna and welcome to your Learn Lab, your one stop for learning. In this video today, we are going to study different methods we employ to study biopsychology. Biopsychology is BPCC 102. It's a very important course subject for first year BA Psychology honor students of IGNOU. And the topic that we are covering here today is from Unit 1 of Block 1. So let's continue with the topic. Stay with me till the end because I'll also tell you the different ways you can remember the concepts and also the previous year questions that has come from this topic. Let us recap quickly. What is biopsychology? Biopsychology is the study of how our brain, body and behavior are connected. Imagine it this way. Brain is our headquarters where every decision of our body is taken. How you walk, how you talk, how do you breathe, eat, whatever task you do at any given time is decided by the brain. Okay, so brain is the headquarters where all decisions of your body are being taken. And what is our body? Our body, all the parts of our body are the workers who carry out all the instructions which are being issued by the headquarters. So the brain says raise your hands, you raise your hands. Brain says blink, you blink. Brain says breathe, you breathe. So parts of your body are the workers. And behavior is the action that we see happening from outside. So when you're looking at something, what you see from outside is the outcome of all the tasks that is being done by the workers as per the instructions of the headquarters. So that's how you can understand the role of brain, your body and the behavior. Your brain tells your body to do something, and when the body does something, what you see from outside is the behavior. So, biopsychology is the study of how the brain, body, and behavior are connected. Brain has been a big mystery for all the scientists who have been trying to study it for a very long time. And since it's already inside our head, protected by the skull, it's very difficult to know how it functions. But still, our scientists have devised different methods to study biopsychology. And these different techniques can be categorized into three simple categories. The first one being looking at the structure. So here we have techniques that we use to look at the structure of the brain or how the brain looks like. It's like how a detective might look at the photo of a city's map to find out where different establishments are like where is the bank or where is the city council or where are the different offices. Similarly, when we look at the brain, we get different parts of the brain where different types of functioning happen. So the first technique that we have is first group of technique we have is looking at the structure. This include histological methods and neuroimaging. We look at these techniques in detail in our coming slides. Going to the second, we have looking at the activity. We have a set of techniques which we use to look at the activity of the brain, where we see which part of the brain lights up when we do different types of tasks. For example, when you are writing something, a particular part of the brain must be active. When you are reading something, it would be a different part of the brain that lights up. When you are thinking or when you are sleeping or when you are walking, for each activity, there would be a separate area of the brain that lights up or is active and helping you do that task. And we use psychophysiological recording to look at the activities of the brain. The third method we have is changing things. Here, what we do is we change or disrupt or destroy different parts of the brain one by one and see what happens. It's like, blocking a road and seeing how the traffic reroutes or where the choking happens or where all the jams happen. So similarly, here what we do is there's a very sensitive technique also because you are essentially destroying a particular part of the brain. And you see that when you destroy that particular part, what kind of functions are being affected, which kind of functions the organism is not able to do, or if there is any function which is being changed, like uh, organism is doing it really quick, or there is a mistake in the whole process. So you can study all the changes that is happening in the behavior when you remove a particular part of the brain. So you can attribute that particular function to that part of the brain, that this part of the brain is responsible for these kind of actions. And under changing things methods, we have ablation method, chemical stimulation, electrical stimulation, and stereotaxic lesions. Let's look at the methods one by one. The first category we have is looking at the structure. And under that, we have histological methods and neuroimaging. First, let us look at histological methods. What is histology? Histology means studying tissues under a microscope to see their tiny structures. So, you know what is tissue, right? Tissue would be a group of cells in your body that would be doing a particular function. Cell is the smallest unit of your body. And when a group of cells come together to do a particular function, we call it tissues. So, in histology, we study these tissues under a microscope to see their tiny internal structures. And this helps the scientist to see what a brain cell looks like, their shapes, connections, and any damage or changes. So this is like zooming into a Lego castle, okay? If you have made castles with those blocks, the Lego blocks or the simple blocks also. So once you have made a castle, maybe someone else comes and they are impressed with your castle and they want to zoom in to see how each block or brick is connected. So this is something similar to what we do in histology. We'll be looking at the tissues to see how they are arranged together to do a particular function. So why is this method important? It helps to identify different types of brain cells, what they look like. It helps to find if there is any injury or changes in the brain after an experiment. Okay. Here, neural tissue is very fragile and disintegrates under normal condition if taken out. So, how can you study tissues if they are so fragile? Neural tissue is very fragile. If you take it out of your brain, it dies immediately, and then it won't be able to show you all the parts. You won't be able to study it under a microscope. So what you do? It needs special treatment before any microscopic examination. Neural tissue, after you take it out, you have to give it special treatment so it does not get disintegrated, and you can still study it under a microscope. Let's see what are those specific treatments. Histological procedures. The first one is fixation. What is fixation? It's like you are fixing the brain or preserving it to prevent its decay. We said that the neural tissues are very fragile, right? So you want to preserve it and prevent any decay. So for that, what we do is we do fixation. Fixation is the preservation of the tissues by cleaning and dipping it in a chemical fixative solution, which would be formalin. So if you have been to zoology labs or plant labs, you might have seen in those big bottles there would be a solution, and in those there would be different specimens. Like maybe it might be different animals or different plants. So that whole process is called fixation. And that fluid that you see inside those jars would be mostly formalin. Okay. So here for studying biopsychology also, we preserve the tissues by cleaning it and then dipping it in a chemical fixative solution, which in most cases would be formalin. There is another method that you can use, which is freezing. So in freezing, you harden the tissue by dipping it in sugar solution and then freezing it at a very low temperature. So this also hardens the tissue sample that you have collected for your study. Another technique that you can use is embedding. Embedding means here also you are hardening the tissue, but by embedding and covering it up in paraffin or nitrocellulose. This is like, you are pouring wax over something and the whole thing gets embedded in the wax. If you know, this is something similar. Paraffin or nitrocellulose also works something similar to wax. So you pour it over your sample, you let it solidify, and you'll have your sample embedded in that solution. Okay. The next step we have is sectioning. Sectioning is you cut your embedded or hardened sample into very thin slices. So when I say very thin slices, The thickness would be, something like your single strand of hair. So if you look at your single strand of hair, imagine how thin it is. So the section that you generate from your hardened sample would be as thin as a single strand of your hair. And for this, a special tool called microtome is used. Okay, so microtome will give you very thin slices of your sample. And once you have got these different sections, you will be arranging these sections on a glass slide and then you will stain it. What is staining? Staining is a process of adding color to make different parts of your tissue stand out. When you look under a microscope, you will see different parts of the tissue stained in different colors. So for that you will be using different dyes or stains. Which are those dyes and stains? Here I have got a few examples. You have Nissl stains, which is used to color the cell bodies. Cell bodies means the bodies of the nerve cells or the neurons. Then we have myelin stain for myelin sheath. This is also another part of your nerve cell which we will study later. And we have Golgi Cox stain for neurons. So in histological methods, the order of processes that you need to remember is you first fix it or harden it, then you slice it into or chop it into super thin slices, and then you add stain so that you can give color to different parts of the tissue, and then the last one you zoom it into a microscope. You use a microscope to observe it. This is what is histological methods. Again, you fix it, you harden it, you cut it or section it, you stain it and observe under a microscope. The next technique that we use to study the structure is neuroimaging. The first one was histological methods. Second one is neuroimaging. What is neuroimaging? It's also called brain imaging. You are imaging the brain here. And this is where we use those huge machines like MRI or CT scan to take detailed picture of the brain while it's still inside the head. So you can study the brain while it's still inside the head, or you can take the brain out and study it, which is not ethically promoted technique. So we have this nicer by alternatives where you can use these machines like MRI and CT scan to still study the brain inside the head itself. This is why it's called a non-invasive technique. Invasive techniques would be those wherein you insert something into the brain, make a change in the brain, or take the whole brain out of your skull. And in non-invasive technique, your brain is not affected at all. It's kept intact. You will be attaching it to machines from outside the skull. It's not invasive. Okay. And neuroimaging techniques can be of two types. The first one is structural imaging. It helps in diagnosing any structural injury or disease in the brain, such as tumor. The second is functional imaging. It diagnoses errors in brain function. So there are two types of neuroimaging, one where you are studying the structure and the second where you are studying the function. This should be easy for you to understand, I hope. Under neuroimaging, the first technique we have is X-ray techniques. I'm sure all of us have seen X-rays at one point or the other, maybe yours or someone else's. So what is an X-ray technique? X-ray technique work by using radiation to create images of the internal body based on different absorption rates of various tissues. So what happens is that you will be sending X-rays into your body, and some of the tissues will be able to absorb those rays, some will not be able to absorb those rays as good as others. So when you finally get the picture or the print or the film out, what happens is that based on the absorption, you will have images in black and white and different shades of gray. I hope you have seen an X-ray. So you might have seen white, it would be a black film and you will be seeing the images in white or grayish color. An X-ray is like taking a quick snapshot of the inside of your body. It's most commonly used to see bones, but it's not detailed enough to look at soft tissues like the brain. So even though it's a very great technique, we won't use it to study brain and for brain we need more advanced techniques. Okay, so X-rays we generally use to study bones, and if you want to check the skull, it is the bony box inside which the brain is kept. You can use an X-ray. It would give you an excellent image of the skull, but brain is made of soft tissues, and X-ray might not give you a good image of those soft tissues. So you might have to use more advanced techniques. Let's get into those more advanced techniques. We have computerized tomography, which is CT scan. CT scan, the full form is computerized tomographic scan. I'm sure you might have heard about CT scan also at some point from somewhere. What are these? CT scan uses X-ray technology combined with a computer to create detailed images of the inside of the body, including the brain. A CT scan is like taking a series of X-ray pictures and then putting them together to create a detailed 3D image. So CT scan is also similar to X-ray, except that instead of a single X-ray image, you will be taking multiple X-ray images from different angles and different orientations, and you'll be combining all those images with the help of a computer to get a complete 3D image of the structure you are studying. A CT scan is useful for seeing brain injuries, strokes, or tumors, like the structural changes that are happening when there is a brain injury or when the patient has a stroke or if there is a tumor. In all those cases, CT scan will give you a very clear picture of what's happening inside the brain. It gives a better image of soft tissues than an X-ray. Okay, so CT scan is similar to X-ray, except for we are using a computer along with the X-ray machines, and we'll be taking multiple X-ray images which the computer will be collating to give you a clearer and better 3D image of the brain. The third we have MRI. MRI stands for magnetic resonance imaging. It is a non-invasive scan. I told you what is a non-invasive scan. Non-invasive scan means you were not inserting anything into the brain. Brain is not being touched directly. It's a non-invasive scan that uses radio waves and a strong magnet to create detailed images of the brain, including the soft tissue. So MRI is a bit more, gives you a clearer picture compared to CT scan. So X-ray would give you a clearer picture of the skull. It will not give you a clear picture of the soft tissues. So we have a better technique, which is CT scan. CT scan will give you a clearer picture or the 3D image of the brain. And we have MRI, which will give you a much more clearer picture when compared to CT scan. And how does MRI work? It uses radio waves and a strong magnet to create detailed images of the brain, including the soft tissues. So MRI produces detailed images of the brain and the spinal cord. It helps to know blood flow to different active parts of the brain. So whichever part of the brain is active, that part will be receiving more blood flow. So if you study the different blood flow patterns to the brain, you will know which part of the brain is active or more active at that point. MRI also provides a higher resolution compared to other methods. The other methods that we have studied till now, which is X-ray and CT scan. MRI doesn't use radiation like X-ray or CT, so this is safer technique. So radiations, I know we need radiations for all those diagnostic techniques, but it's still not safe for the body. And MRI is a technique which does not use radiations. And what does it use? It uses radio waves and a strong magnet to create a magnetic field, and based on which the structures of your brain and spinal cord are studied. Next we have PET scan. This is positron emission tomography. I'm sure you might have heard all of these terms, but you might not have learnt about it, or you might not know what exactly this is. PET is a very common diagnostic technique that is used to spot tumors in any part of the body. So PET scan is one technique which will tell you if there is a tumor in any part of your body. Full form of PET is positron emission tomography. It measures brain activity by detecting radioactive tracers that are injected into the body. So when you go for PET, what happens is that the doctor or the whoever is the radiologist there, they'll be giving you a bottle of water in which all these radioactive tracers are added. You are asked to drink that water. So what happens is that when you drink that water, this radioactive tracers enter your body and it moves to different part of the body. And once your body has absorbed all those radioactive tracers, you are asked to enter a PET machine, the scanning machine. And that scanning machine is able to look at all the in which all parts the radioactive tracers have been absorbed, and based on that absorption the doctor can identify if there is a tumor where there is a different or not normal pattern of absorption, and they'll give you a detailed image also of all the absorption patterns. So, a radioactive tracer, usually a modified form of glucose. So, glucose is something that all the cells of our body uses for as fuel. Like every single cell in our body survives due to glucose. So, a radioactive tracer or a modified form of glucose is injected into the bloodstream. The tracer is absorbed by the active brain cells which use more glucose. So the part of the brain that is more active will be using more glucose. As the tracer decays, it emits positron that collide with electrons producing gamma rays. This is a technicality that even if you don't remember it is okay, because it's already a radioactive tracer that is being injected. It will be emitting radioactive radiations. All you need to remember is the radioactive tracer, which is a modified form of glucose, is injected into the body, and after it is absorbed by the cells or the nerve cells in this case, it emits radioactivity. Your body at that point will be giving out radioactive radiations. And then a scanner detects these gamma rays and creates images showing brain activity. Okay, so once your body is scanned at that point, the scanner will detect all these radioactive radiations coming out of your body and create an image that shows the brain's activity. It reveals areas of increased or decreased metabolic activity, which can help diagnose conditions like tumors, seizures, and Alzheimer's disease. Okay, so wherever there is more activity, more glucose would be taken up. So in case there is a tumor, what is tumor? Tumor is where a cell would be growing uncontrollably. A cell would be multiplying uncontrollably. So when there is uncontrollable cell division happening, there would be a higher metabolic activity at that point. And where there is a higher metabolic activity, more glucose would be taken up. If more glucose is being taken up, because this glucose is radioactive, there would be more radioactive radiations being emitted out at that point, which the scanner will be able to identify. And this is how PET works. Next we have fMRI or functional MRI. So in simple terms, fMRI helps us to see the wiring of our brain, the wiring or the connections or how the nerve cells are connected to each other, which allow different parts of the brain to communicate with each other. So functional MRI, it helps us to see how different parts of the brains are connected to each other and how they communicate with each other. fMRI relies on the principle that when a brain region is active, blood flow to that region increases, bringing more oxygen-rich blood. We have seen the same principle being used in PET scan also, where more the activity, higher the blood flow, higher the glucose intake also, and hence higher the radioactivity. Here also, functional MRI relies on the same principle that when a brain region is active, blood flow to that region increases, bringing more oxygen-rich blood to the region. The fMRI machine detects these changes in blood flow and oxygen levels by using a strong magnetic field and radio frequency pulses. So, if you remember MRI was not using any radioactivity, or any radiations. It was using a magnet as well as radio waves. Here also, we'll be using a strong magnetic field and radio frequency pulses to detect the changes in blood flow and oxygen levels. Okay. These changes in blood flow and oxygen levels are used to create images that show which part of the brain are active during a particular task. So this technique comes under functional neuroimaging, where you are studying the function of the brain. Okay, so the changes in the blood flow and oxygen levels are used to create images of the brain, which is working or which is active at that particular time period. Next we have diffusion tensor imaging. What is diffusion tensor imaging? The name sounds pretty complicated, but it's not. It's a type of MRI that looks at how water moves through the brain to map the connection between different brain regions. You know that our human body is made of water. We have a lot of water in us. If you get dehydrated, you will die. Brain also need water to survive. So diffusion tensor imaging or DTI, it's a type of MRI that looks at how the water is moving through the brain to map the connection between different brain region. It helps to visualize white matter pathways and assess brain injuries like traumatic brain injury. So our brain has two types of matter, white matter and gray matter. This we'll study later. So maybe at that point you will be able to understand this more. For now, all you need to know is that DTI or diffusion tensor imaging helps to visualize the white matter pathways. And this will help to study brain injuries like traumatic brain injury. It gives an idea how different parts of the brain are connected to each other. So this is also a part of the functional MRI and structural MRI also. So this will help you to see the white matter pathways. It will help you to assess brain injuries. All the while it will also tell you how different parts of the brain are connected to each other. Next we have transcranial magnetic stimulation or TMS. Transcranial. Cranial means your skull. Trans means across. So you are using magnetic stimulation to study across the skull. So this is a non-invasive technique again, because whatever is happening outside the skull. So you are having technique which functions outside the skull to study what is happening inside the skull. So that's why this is called transcranial magnetic stimulation. It is a non-invasive procedure that uses magnetic fields to stimulate specific areas of the brain. So here, what are we using the magnetic field for? We are using the magnetic field to activate or stimulate certain areas of our brain. How do we do that? A coil is placed on the scalp and a rapidly changing electric current is passed through it, generating a magnetic field. So you place a coil on your head and a rapidly changing electric current is passed through it. And when the electric current passes through the coil, it generates a teeny tiny magnetic field. This magnetic field interacts with the brain tissue, inducing electric currents in the neurons. So this magnetic field will induce electric currents in the neurons inside the brain tissue. The induced electric current can either excite or inhibit the neurons depending on the frequency, intensity and duration of the magnetic pulses. So all our neurons which are connected to each other communicate with each other using electrical signals, and these electrical signals are automatically generated by the neuron. Here what we are doing is we are externally giving a electrical current to the neurons, and this can either inhibit the neuron or excite the neuron. So based on whatever action happens on the neuron, it can either get activated or it can either get inhibited. You will see a change in the behavior. Okay. By modulating neuronal activity in specific brain regions, TMS can influence cognitive processes, mood, and other brain functions. So what we are doing here, we are having coils attached to our scalp. It's going to generate a electric current. This electric current will generate a magnetic field. This magnetic field will affect the brain, and how it is going to affect the brain? It will induce electric currents in the neuron. And when currents are passing through the neurons, it can either get activated or inhibited. And based on whatever happens to the neuron, it can influence your behavior, the cognitive processes, mood, or other brain functions. So these are the different neuroimaging techniques that we use to study the structure of the brain. I have a summary image here for neuroimaging. What are the different neuroimaging techniques that we studied? We studied X-ray. It uses radiation to view bones. It will not help you to see the soft tissues of the brain. So we need more sophisticated or more sensitive techniques to study the brain. Next we had CT scan. It is same as X-ray, but it uses multiple X-ray images to create a more 3D, more detailed 3D image of the brain. Then we saw MRI. It gives you a detailed image of the brain structure using magnetic fields and radio waves. Then we had PET scan, where you are using radioactive tracers to measure the brain activity. Then we saw functional MRI. It detects changes in the blood flow. Then we had transcranial magnetic stimulation, which stimulates brain using magnets. Then we have diffusion tensor imaging, which maps brain's connections. So that's all about neuroimaging. So we have studied two techniques to study the structure of the brain. One is histological methods and second is the neuroimaging. Next we have techniques that we use to look at the activity of the brain. The technique that we use to study the activity of the brain is psychophysiological recording methods. What is this? So imagine your body as a radio station. So what do radio stations do? It produces signals which can be picked up by different types of receivers. And here in the case of the brain, these signals tell us about what's happening inside the brain and how you're feeling. Okay. It is a non-invasive method, which means your brain is not directly being touched or affected. No surgeries, no insertion, all the studies happen from outside the skull. So, and yeah, that's what is the meaning of a non-invasive method is studied from outside the skull and non-insertion, no insertions. Let's see different techniques that come under psychophysiological recording methods now. The first one is EEG. I'm sure we have all heard about ECG, so we might also have heard about EEG. EEG stands for electroencephalography. Okay, so what does EEG do? It records the electrical activity of the brain using the electroencephalograph machine. As I told you, the cells of the brain or the neurons communicate with each other by generating electrical signals. So, all of our brain, there is a continuous electrical activity happening between the neurons. And the EEG, it records this electrical activity using the machine which is called electroencephalograph. Okay. And the recorded activity is called electroencephalogram. You might have seen those papers with those wavy forms. That is called electroencephalogram. It is the recording of the electrical activity of the brain. So to simplify, think of the brain as a busy messaging center. It's constantly sending out messages in the form of electrical signals. And EEG is like a device that listens to the brain's radio station and records all the chatter that's happening inside the brain. How do we do EEG? Electrodes are placed on different points on the scalp of the patient and the brain waves are recorded. So if you see here in the image, there are all these electrodes that are attached to different parts of your scalp, and then all these electrodes are attached to a computer or a reader, and electrical activity of the brain is mapped by this recorder or EEG reader. It helps in studying different states of consciousness, attention, and responses to stimuli. So, if you are sleeping, EEG tells you which parts of your brain are active and which is not active. How? Based on where all the electrical activity is happening and where it is less. Similarly, if you are studying something or reading something, if you are connected to an EEG machine, it will tell you while you are reading which parts of your brain are active or where the electrical activity is more. So that was all about EEG. Next we have magnetic encephalography or MEG. The first one we saw was EEG which is electroencephalography. The second we have magnetoencephalography, which is MEG. It's a non-invasive technique, which means all the study happens outside the skull. This one employs highly sensitive magnetometers to map brain activity by detecting the magnetic fields produced by electrical currents in the brain. So I told you that brain functions by creating electrical signals which are used by the neurons to communicate to each other. So when these electric signals are generated, the brain is also able to create a tiny magnetic field. Okay, so brain, in addition to creating electrical signals, it also creates tiny magnetic fields when it works. And what does MEG do? The MEG works like a special metal detector, okay, that pick up these weak magnetic signals. Have you seen in maybe in any TV shows or anywhere where metal detectors are being used? Those kind of handy machines, they are scanned over the surfaces, and whenever it detects a metal, it starts beeping. Okay, so imagine MEG also works something like that. It will be scanning your body and it will be picking up these weak magnetic signals which are generated by the brain. And it helps us to find out from which part of the brain these signals are coming from or where exactly all the action is happening in your brain. It's a tool which is used to understand brain function and identify the source of epileptic seizures. Okay, so this is the principle of magnetoencephalography, that it will be picking up all the weak magnetic fields that are generated by the brain while it is working. And the scanner, the magnetometer, it is highly sensitive. It can detect all these small magnetic fields and it finds out where all the activity in the brain is happening. So if someone have an epileptic episode, MEG will be able to find out which part of brain is generating these seizures. It will help in treatment also. So that was all about magnetic encephalography. After EEG and MEG, we have EMG. This is electromyography. Okay, so it might be a bit tricky to learn all the names, but you might have to put some effort into learning these names. Like first one we had EEG, which is electroencephalography. Then we had MEG, which is magnetoencephalography. Third we have EMG, which is electromyography. What is EMG? So imagine your muscles as workers in your body, okay? All the muscles in your body are workers which do different types of work. And for these workers to work, they need instructions from the brain. So brain will tell move, so the legs will move. Brain will tell your hands to pick up something, it will pick. Brain will tell your heart muscles to contract and relax, it will work accordingly. Okay, so all the instructions for your working of your muscles come from the brain. And EMG, it is like a listening device. It listens to all the instructions that a brain is sending out. And how does the brain send out these info instructions? In the form of electrical signals, okay? And these signals which are going into your brain are picked up by this EMG recorder or reader. So for example, when your brain tells your legs to bend, to walk, EMG can detect that muscle activity and tell how much effort you are putting into doing that particular function or action or that movement of the muscle. Okay. So what is electromyography? It is a diagnostic procedure that evaluates the health of muscles and nerves that control them. It evaluates the health of the muscles and the nerves that control these muscles also. Okay. It measures the electrical activity of muscles both at rest and during contraction using electrodes attached to them. Here also electrodes are attached to different muscles of your body which are under study, and whenever there is an instruction or electrical signal coming from the brain to these muscles, EMG will record these signals. It is used to diagnose neuromuscular disorders. Neuromuscular disorders are those disorders which happen due to, what do I say? If the communication between the brain and the muscle is not proper, it would cause uh, disorder, or not exactly disorder. So this would be something like your nerves will not be able to bring in messages to the muscles, and because there is no instructions coming up, the muscles will not be able to perform the task it is supposed to do. So this is called neuromuscular disorders. So MEG can help to diagnose neuromuscular disorders, and it can also help differentiate between nerve and muscle problems. So whether a problem is caused due to muscles or it is due to the nerves, EMG will be able to diagnose that also. Next we have electrooculography or EOG. So your eyes, it is moving around constantly. Okay. And this movement of eyes is also controlled by eye muscles. And these muscles also send out electrical signals to the brain, and brain also sends signals to these muscles. So EOG, what it does is it detects these signals that are coming in from brain to the muscles and the eye muscles and the eye muscles to the brain, and it can help track exactly where your eyes are looking. So it's like a camera that can detect your eye movement when something attaches your attention. So maybe a passing vehicle or a beautiful flower catches your attention, and then when it does that, your eye moves to that object, and this movement is detected by EMG, that your brain has sent your eyes a signal कि there is a beautiful flower, look at it. And when that message comes from the brain, the muscles, it makes your eye move to that particular region. So this tells how much the connection between the function between brain and eye muscles are working. So electrooculography or EOG. So we had EEG, we had EMG, we had MEG, now we have EOG. It records eye movement. How do we study this EOG? Again, electrodes are placed near the eyes. Patient is asked to move their eyes between fixed points in alternating light and dark conditions. The potential difference created by the back and front of the eyeball is measured. So what is potential difference? So the electrical signals that are being generated at the back of the eyeball and front of the eyeball. The difference between these electrical signals, the potential difference, is being measured by EOG. I hope you understood that. The last technique we have under psychophysiological recording method is electrodermal activity or EDA. A lot of abbreviations we have learned, right? So we have one more here. This is EDA or electrodermal activity. So how does this work? When you're nervous or excited, what happens is sometimes there are these sweat glands in your skin. This will produce a little amount of sweat. For some people, it would be so less that they may not even notice it. But for some people, the sweating will be very profuse. Okay, just a tiny trigger, a nervousness or excitement will trigger the sweat gland. So EDA, it measures these tiny changes in your skin's electrical conductivity when that happens. Whenever you are excited or nervous, this EDA will catch these changes in electrical conductivity, and it helps to check your skin to see if you're feeling stressed, scared, or excited. Okay, so what does electrodermal activity do? It measures changes in the electrical conductivity of the skin due to sweat gland activity. So for sweat gland also to be active, the signal has to come from brain, and the signal comes in the form of electrical signals. Okay. These changes are linked to sympathetic nervous system activity, indicating emotional arousal and physiological responses. So, sympathetic nervous system is a type of nervous system in your brain. Again, this we'll study in detail later. So, sympathetic nervous system, it is responsible for emotional arousal and physiological responses. And these responses would be studied using electrodermal activity. The skin conductance level or SCL, this is the background level, or when you are at rest, when you are not excited, you are perfectly neutral state. whatever the skin's conductance or the electrical activity happening on your skin is the skin conductance level or the background level. And the skin conductance response. This is the response to a situation. When something happens, how your brain responds, how you respond to it. You might be nervous, you might be excited. That kind of response, when that happens, the changes in the electric conductance of your skin is the SCR or the response to a situation, are the two indices measured. So electrodermal activity measures the background level of the skin conductance when you are at rest and there is no emotional activity going on in your head. And then it also measures the skin conductance response. This is the electrical activity which happens in response to a situation. Okay, so that was electrodermal activity. I'll give you a trick to remember. I don't know how much it works, but it did work for me. So you can check, or maybe you can create your own mnemonics, which will help you to remember all these abbreviations and their full forms. So what did we just complete? We completed psychophysical recording methods. These are techniques which are used to study the functions of the brain or the activities of the brain. Before that, we studied techniques or the methods which are used to study the structure of the brains. Which were the two methods that we studied? Histological methods and then neuroimaging. So for psychophysical recording methods, this is the trick that I have. Imagine you're playing a video game, okay, and you were really focused in it. So in this case, your brain is active. When your brain is active, how do we measure the activity? Using EEG and MEG. So we attach electrodes and it will tell where the maximum activity of the brain is happening. Okay. Your brain is active, your eyes are scanning the screen. Okay, video game means your eyes are continuously scanning the screen also what's happening where. So what do we use to measure that? EOG. Okay. Then your muscles are tense. You're using your hands, your muscles are tense, you have to kill all the enemies. So how do you measure that? Using electromyography or EMG. Okay, this was electrooculography, EOG. Okay, what was EEG? Electroencephalography. What was MEG? Magnetoencephalography. Okay, so you have to study the activity of your brain. You use EEG and MEG, and then your eyes are continuously scanning the screen, which you can study using EOG, and your muscles are tense, which is studied by using EMG, and then you might sweat a little from the excitement or stress. Okay, and this will be studied by EDA. By measuring all these signals together, we can understand exactly how your body reacts to the game, even when you don't realize it. Okay, so that was all about psychophysical methods. Next, we have the next category, that is changing things. Changing things, what do we say? We said that we will be making changes in the structure of the brain and see what happens when those changes are made. Okay, so the first method under changing things is ablation method. In ablation method, what you do is you intentionally remove or destroy specific areas of the brain, and then you see what activity is not happening in the body because of that removal. What are those functions which the organism or the animal is not able to do because of the absence of that part of the brain. It is a very invasive method because you are penetrating the skull and all your studies are happening by making changes to different parts of the brain. Changes that occur in the behavior due to the destruction or removal of the tissue are studied. An important term that you need to know here is lesions. What are lesions? These are any damage that happens to the areas of the brain due to accidental injury or deliberate ablations. So when you are making that cut or making an injury to any part of the brain intentionally or an unintentional accidental injury, the resulting change is called lesion. Okay. A caution that you need to know here is that Parts of brains are connected to each other. So removal or destruction of one area can affect other areas also. And this might be very difficult to identify too. So you have removed a part of a brain of the animal you are studying and you saw a particular change in behavior because of that. But point is this could be because of some other part of the brain which got affected by removal of the part that you actually uh, took away. You'll never be able to know that this is not because of the part that you have removed, but because of a different part of the brain. So you have to be very careful while devising experiments using ablation methods. And sometimes the animal recovers from the brain damage partially. Functions of the damaged parts of the brain may even be covered up by other parts of the brain. So as I said, brain is a very dynamic and mysterious organ we have. So you'll never know what exactly is happening inside the brain. It could be that the animal has recovered from the damage, or maybe the functions of the part that you have removed is being carried by some other part of the brain. Okay, so in which case you will never know what kind of function was affected by the removal that you have done. So ablation method is a very invasive method and it's a very sensitive technique, and every kind of ethic, whatever there are ethics in animal experimentation, all those are applicable when you try ablation methods to study the brain. We have different methods of ablation. So ablation is basically causing lesions. So we have different methods of causing lesions in the brain. And the first one we have is aspiration. Aspiration works like sucking. Like maybe you might have seen the vacuum cleaner. It sucks up all the dirt from wherever you're cleaning. So aspiration also works like that. It's like using a tiny vacuum cleaner to gently suck out a part of a brain or a tiny piece of the brain. Okay. So scientists do that to see, uh, you know, what changes happen when this part is missing. This is all about ablation method we know. So aspiration also does the same thing. It gently sucks out a part of the brain, and then you can study what changes are happening because of removal of that particular part, or what functions stop or slows down because of it. And how do we do? A fine needle attached to a syringe is used to remove fluid or tissue samples from a specific area of the brain for analysis. The samples are spread out on a slide and examined under a microscope. This is minimally invasive and there is minimal destruction of surrounding area. There is destruction, but it is minimal. And this is used to remove cortical cells to study the areas underneath. Cortical tissues would be the outermost tissues, which is closer to the skull. So if you want to remove the outermost part so that you can study more about the inner parts, you will use aspiration methods to remove those cortical areas. I hope you understood aspiration. It's pretty simple, works like a vacuum cleaner. You suck out a part of the tissue that you want to remove using a fine needle attached to a syringe, and then you study the sample under the microscope. And you will also see the change that has happened in the behavior of the organism that you are studying. Next, we have radio frequency. So the first one, aspiration, is like using a vacuum cleaner. Radio frequency is like burning that part using heat. Okay. Have you seen soldering iron? Radio frequency is like using a soldering iron to burn a spot or a part of a wire. So how do we do radio frequency? A wire is placed on the tissue that needs to be studied. Okay. This wire is connected to a radio frequency device. The device produces an electric current. And when electricity passes through that part, heat is generated by the electric current, which destroys the tissues to which the wire is attached. Whichever tissue the wire is attached to or the wire touches, those parts are burnt or destroyed by the heat generated by the electric current. The duration and intensity of the current determines the size and shape of the lesion. So if you are passing the electric current for a longer duration and the intensity is high, then the damage would be more. So the size and shape of the lesion would also be bigger. Okay, so radio frequency, we are using heat to destroy the tissues that we are studying. First one was aspiration. You are sucking it out. Radio frequency, you are burning it. Third, we have knife cuts. So here, this is like snipping up. Okay, you are snipping or cutting a wire with a knife. This is similar. Scientists make careful cuts just to cut the connection between brain areas without destroying the whole area itself. So they are using a knife to make a cut that will create a what do you say, that will sever the connection between two parts of a brain. So then they study what happens when intact parts are not communicating with each other. Okay, for example, we have a right part of the brain, we have a left part of the brain. If you remove the connection between the two, you never know what is happening because there are no communication between the two parts. For those kind of studies, you use a knife cut. This is minimally invasive surgical techniques that uses a 3D coordinate system and imaging to locate and make cuts to subcortical regions. So you're using a highly sensitive 3D coordinate system and imaging so that you can see inside the brain and make the cut in exactly where you want the cut to be without disturbing other parts or other areas of the brain. So this disrupts the neural communication between different parts of the brain. So here no lesions as such, no destruction, just severance of the communication between two parts of the brain. Again, first we learnt aspiration, sucking out. Second we learnt radio frequency methods where we are burning the tissues. Third we have knife cuts where you are severing the connection between two parts of the brain. Next is cryogenic blockade. And here what you do is you are freezing the part. Okay. So this is a method to induce temporary lesions. A cryoprobe. Cryoprobe would be a probe or a needle which would be like cold, very cold. It's much cooler than the temperature would be much cooler than what the cells can endure. So the cell freezes. When it freezes, it doesn't die. It just stops functioning. Okay. You're not killing the cell. You are just inactivating the cell by giving it a very low temperature. Okay. So a cryoprobe is placed in the brain. A coolant is sent to the area under study through the probe. A coolant would be any chemical that would reduce the temperature of that area. Coolant reduces the temperature of the area and the neurons stop sending messages. Again, remember, you are not killing the neurons here. You are just inactivating them so they are not sending messages anymore. So this is a very temporary process or the on reverting the temperature, the neurons would start functioning again. There is no structural damage involved. This is one of the very safe invasive methods where you are using a cryoprobe. You can see it in the image also what a cryoprobe looks like. And through this probe, you will be sending a coolant. This would be a chemical which reduces the temperature of the area. And when the coolant reaches the area that you are studying, it freezes the neurons because of which it stops sending messages. And then you can see the difference in behavior of the organism or what kind of functions are affected by those neurons not functioning. And here you are not creating any structural damage. Whatever is happening, it's a very temporary process. Whenever the neurons are warmed up, it starts working again. The last technique under ablation, we have nerve poison, and nerve poison is a chemical. So we have used electricity, we have used heat, we have used cold. Now we use a chemical. Okay, so here there is a chemical attack on the brain. These chemicals kill only the neurons that you are targeting without hurting any other cell or blood vessels nearby. And how do we use? You use a glass tube called cannula. It's used to introduce nerve poisons such as cyanic acid or ibotenic acid to the area under study. Okay, you will be using a cannula or a glass tube to introduce nerve poisons, and examples of the nerve poisons are given cyanic acid or ibotenic acid to the area under study. And what do these poisons do? These poisons create lesions by destroying the cell bodies in the area. This would be killing all the neurons in that area, and in that way it will be creating a lesion. There is only a partial destruction, as the any other cell that is not targeted will not be affected by the poison. Okay, so that was all about ablation methods. So let's summarize the ablation method quickly. First, we saw aspiration, where suction is being used to remove a part. Then radio frequency lesion, where the local tissue is being destroyed using heat. Then we used knife cuts, where you are severing the connection between the areas. Then we saw cryogenic blockade, where you are cooling the area temporarily and inactivating the neurons. Lastly, we saw the nerve poison, which is a chemical agent that kills the neurons. After ablation, the next technique we have is electrical stimulation. So what are we studying? These are the methods to create lesions in your brain so that you can study the changes which are happening. Okay. Electrical stimulation. Imagine your brain is a huge network of light bulbs, okay, connected by a wire. So generally your brain decides which bulb it wants to light up at any particular time. And based on that, it will send the electric signal to only that bulb. Okay. And then what happens? Only that bulb is lighted. And because of that, a particular part or area will be lighted up. So in case of your body, what happens is that based on that electrical signal, you will be performing different activities like moving, thinking, feeling. Okay. So in electrical stimulation, you are sending out a small or controlled spark of electricity to a specific part of your brain to turn it on and see which part of the body or which part of which function is affected by it. That was a simple explanation of electrical stimulation. Let's see what it is. It is a technique that uses electrical currents to influence brain activity. It involves directly stimulating brain tissue through electrodes implanted in the brain or indirectly by placing electrodes on the scalp. Sorry. So you can insert the electron into the brain or you can just attach the electrodes outside on the scalp. Either way, you will be stimulating different parts of the brain tissue. You will be sending an electric current. This will activate different parts of the brain. Electrical stimulation changes the resting membrane potential and an action potential is generated. Resting membrane potential and action potential are important terms when it comes to activity of the brain. Again, this is the concept that we'll study in detail when we learn neural conductance or conduction inside the brain. So in simple terms, what you need to understand is that I said the whole function of brain happens due to electrical activity. So when the brain is at a resting stage, there is a resting electrical activity. And when there is a stimulus or there is an excitement, this electrical activity increases. That is action potential. So electrical stimulation changes the resting membrane potential or the resting electrical activity and generates an action potential or it increases the electrical activity, resulting in a behavioral change depending on the area of the brain that is stimulated. This is used for research purposes or to treat neurological or psychiatric conditions. You might have seen electrical shocks being given to patients, right? Those are electrical stimulations, but we don't use that for research and all. But we use, but with the purpose of activating a part of the brain and see which function is affected by that stimulation. Two types of recordings are done using electrical stimulation. First is a chronic recording. This is a recording made over a longer period of time. It involves implanting devices that allow for long-term monitoring of brain activity, potentially for months or even years. So what you do is you implant an electrode into the brain of the animal under study. You let the implant remain there for longer period of time, maybe for months or years, to record the activities of the organism. Example, you want to record the brain activity while the organism is recovering from a brain injury. So it might take months or even years for the animal to recover from the injury or the lesion that you have created. And while that recovery happens, whatever brain signals or brain activity is happening, you want to study. And for that, you use a chronic reading method, which is a long-term reading method. Then there is an acute reading method. This is exactly opposite of the chronic reading. This is a short-term recording done under anesthesia or with a minimal brain exposure often involving a single session. You will be either attaching the electrode on the scalp or inserting it while the animal is under anesthesia, and then you will be studying the brain activity. Then you will remove all of those electrodes and everything, and then you can revive the animal. The animal is safe and go on living. Example, to study a particular nerve pathway. Maybe you want to study sleep. When you want to study sleep, you would like to put the animal under sleep and then see which part of the brains are working. This is acute recording or short-term recording. The electrical activity output can be obtained in three ways. Okay, so when you are doing the electrical stimulation, the outcome or the electrical activity that is happening in the brain, this can be studied using three ways. The first one is a device called oscilloscope. You can see this image here. This is how an oscilloscope will look like. It displays electrical recording per unit time. Maybe in 1 minute, how much electrical signals are being generated. You will see wave like this. Okay. It has a cathode ray tube on which the electrical activity is displayed. Basically the screen, okay, on which the electrical activity is displayed. It can record the activity of a single neuron also. It can record the activities of a whole tissue where there would be many different neurons, or it can detect the activity of a single neuron also. After oscilloscope, we have ink writing oscilloscope. This is also called polygraph. It's an older type of oscilloscope or oscillograph. Here what happens is that there is no computer or screen attached. It has a pen arm attached to a coil, and this pen arm would move according to the electrical signal and trace a graph on the paper. So as the pen moves, it will draw a graph on the roll of the paper. It's a very older technique, very rarely used nowadays. It helps to detect any tumor in the brain, epilepsy, and sleep patterns. So first you had oscilloscope where the screen will show all the recordings. Next we have an ink writing oscilloscope where there is a pen arm which will draw the waves as per the electrical activity being traced or recorded. Third, we can use a computer. It helps to display, store, and analyze information obtained by the recording of the electrical signals. So, here we are displaying, here we can display, store, and analyze information obtained by the recording of the electrical signals. So, that was electrical stimulation for you. Next, we have chemical stimulation. Here, as the name suggests, we are using chemicals to create lesions. And lesions are what? Lesions are the areas where the tissues are being destroyed. So, use of chemicals to influence the activity of neurons and other cells in the brain and body. This is chemical stimulation. Chemicals can be administered by directly injecting into the brain through a tube inserted into the stomach. From the stomach, it will be absorbed by the blood, and the blood will take this into the brain, or injecting into a vein or a fatty tissue in the body. So these are the different routes through which you can make sure the chemical reaches your brain. First, you can directly inject into the brain. Second, through a tube inserted directly into the stomach. From the stomach, it will enter the blood. Blood will take it into the brain. Then the third is you can inject into a vein or a fatty tissue. Again, it will enter the blood or the lymph, and then it is taken to the brain. Different techniques used in chemical stimulation. The first one is 2-deoxyglucose technique. This is used to study local cerebral glucose utilization. Local cerebral glucose utilization means utilization of glucose by local parts of the brain. I told you, when a part of the brain is working or active, there would be more metabolism happening in those cells. So there would be more blood flow to that part, and the blood would be taking more glucose to the part, and the cells will be utilizing this glucose to generate energy for its function. Okay, so 2-deoxyglucose techniques is used to study this local glucose utilization by injecting a radioactive analog of glucose. Where did we study the radioactive analog of glucose before in this video? We used it in PET scan, where I told you we'll be injecting a radioactive analog of glucose into the body, and then based on wherever the activity of brain is more glucose would be taken up. Where there would be more glucose, there would be more radioactivity, which would be detected by the PET scanner. Okay, here also we are using the same principle. There is a radioactive analog of glucose which is called 2-deoxyglucose, 2-DG, into an organism, and then we measure its distribution in the brain. Okay, 2-DG is injected into the animal and incubated till the chemical reaches the brain and is absorbed. The brain is then removed. Okay, this is a very invasive technique because here animal will be killed to remove the brain. Then thin sections are made and studied using autoradiography. So autoradiography is a technique which is used to detect radioactivity coming out from the sample. In this section that you have made, wherever there is radioactivity, this autoradiograph will show your brain's image and it will show dots wherever the radioactivity is there. Okay. Slices are coated with photographic emulsion and then developed as a film. Neurons that take up the chemical appear as distinct spots. So let me just quickly revise this. There is a radioactive analog of glucose which is called 2-deoxyglucose. We will be injecting that into the organism and then wait for it to reach the brain. And after it reaches the brain, you will be taking the brain out from the animal skull. We will be making thin sections of the brain, and after you make thin sections of the brain, you will study it using autoradiography, where you will be able to identify which all area is displaying radioactivity. So then when you are doing autoradiography, what you do is you coat these slices with photographic emulsions so that you can get the final output like a film. You might have seen those old cameras where you put in a film and then develop the images. This is something very similar to that. Okay, so neurons that take up chemical appear as distinct spots. The next technique we have cerebral dialysis. What is cerebral dialysis? A tube is inserted into the animal's brain. Then the animal is engaged in an activity. Okay, then the neurochemicals generated during the activity are collected and studied using chromatography. So here what you do is you are inserting a tube into the animal's brain and then you engage the animal in an activity. Maybe running or maybe trying to solve a problem or learning a trick, something, any activity. And then the brain, while it's functioning, will be generating certain neurochemicals, which we also call as neurotransmitters. Okay. So using this tube, we will be collecting those chemicals, neurochemicals that are being generated, and then we study these chemicals using chromatography. So chromatography is a technique where we separate out these chemicals and each of these chemicals can be extracted and studied independently. Okay, that was cerebral dialysis. Next we have in vivo voltammetry. In vivo voltammetry is a technique that allows for real-time monitoring of neurotransmitter release and metabolism in living organism without the need for sample extraction or sacrifice. So this is also doing exactly what our earlier technique was doing. Here also, there we were inserting a tube and collecting all the neurochemicals. But in vivo voltammetry, it allows you to study the same thing, all the neurochemicals that are being generated while the animal is engaged in an activity, but without inserting any tube. Okay, no need of sample extraction or any sacrifice to remove the brain. This will do everything in vivo or without any invasive procedures. Next, we have immunocytochemistry. This is a technique that uses antibodies to detect and visualize the presence and localization of specific proteins or neurotransmitters within cells. So here what happens is that suppose you want to study chemical which I call suppose A, chemical A. You want to study when chemical A is released by the brain, what is the amount of it released at when the animal is doing different types of activities. So that what you do is you generate an antibody against the chemical A, neurotransmitter, neurochemical A. You create an antibody against it. So what does the antibody do? It goes and specifically binds to this uh, chemical. Okay. Now, if you have a dye attached to the antigen or a radioactive chemical attached to that antigen, wherever your antigen goes and binds to the chemical, it will show a particular color or radioactivity. Okay. This way, you can detect and visualize the presence and localization of specific proteins or neurotransmitters within the cells. How? You are having an antibody which is attached to a coloring agent or a radioactive agent. This antibody will go and bind to the chemicals. In this way, when you are looking at the color or the radioactivity, you will know which parts of the brain have more of those chemicals and what activity is connected to that chemical. Like when an organism does which activity, this chemical concentration increases or decreases in which part of the brain. So this was immunocytochemistry or techniques of chemical stimulation. So last technique we have under chemical stimulation is microiontophoresis. Okay. It uses an electric current to eject small amounts of ionized neurotransmitters from a micropipette, allowing for precise and targeted delivery to specific areas of the brain or other tissues. So, what happens here? Electric current is used to eject small amounts of ionized neurotransmitters. Neurotransmitters are neurochemicals which are naturally generated in the brain. But here you are externally injecting ionized neurotransmitters into the specific area of the brain which is under study using a micropipette. Okay, so this is allowing for precise and targeted delivery of the neurotransmitter to specific areas of the brain, and then you study the effects of these neurotransmitters and other signaling molecules on neuronal activities. So neurotransmitters are naturally produced by the brain, but you want to see that if you provide these neurotransmitters externally to a particular part of the brain, how it affects the activities or the behavior of the organism under study. This is also another technique which is used under chemical stimulation. After ablation method, we have stereotaxic lesion. Okay. Again, this is another method of creating lesion to study the function of the brain. This also comes under changing things, where you are changing the structure of the brain and studying the function. Stereotaxic lesion or stereotaxic surgery is a minimally invasive technique that uses a three-dimensional coordinate system to precisely locate and manipulate brain tissue. Okay, so here you will be using a 3D coordinate system. It would be like a screen or a camera which will help you to precisely locate and manipulate a part of the brain tissue. The identification of the exact location to be studied involves using a specialized instrument or stereotaxic apparatus and a brain atlas to guide surgical procedures. So here we are using a special instrument like a camera to identify or locate the exact location and a brain atlas to guide surgical procedures. The atlas provides a map of the brain, allowing researchers to navigate to specific coordinates and target areas of interest. Okay, so this is a surgical methods, invasive method. We're using a brain atlas and a specialized instrument called stereotaxic apparatus to locate the exact position of the part of the brain which you want to study. So after you have located the region or the part of the brain that you want to study, the animal is administered anesthesia and the head is firmly held in the apparatus. The electrode holder is used to insert the electrode or cannula into the brain. And when the animal comes out of anesthesia, the behavior is monitored. And when the study is over, the electrode can be removed surgically and the animal discovers from the lesion. So here you are using highly sensitive camera to locate the part of the brain which you want to study. This you will do by administering anesthesia to the animal. When the animal is under anesthesia, you will be firmly holding the animal's head in the stereotaxic apparatus, and using the electrode holder, you will be inserting the electrode or cannula into the brain. So this is a very invasive technique. And after the animal comes out of anesthesia, the lesion is already created there due to the presence of electrode or cannula. And then you allow the animal to go about with the daily activities. And you study the changes in behavior, like what changes in the behavior of animal has happened due to the presence of those lesions created by the presence of electrode or cannula. And after you have completed your studies, you can remove the electrodes surgically and the animal recovers from this lesion. Okay, so this was all about stereotaxic lesion. This was a method employed for creating changes in the brain and to study its effect. With that, we have completed all the methods used to study biopsychology. Okay. Next, I have some previous year questions that have come before from this topic. What was the topic? Methods to study biopsychology. Latest in June 2024, the question came, write short notes on the following in about 150 words. This was for 5 marks. And the question was, electrical stimulation to study biopsychology. We have covered this topic. In December 2023, the question came, explain neuroimaging techniques to study biopsychology for 10 marks. We have studied neuroimaging. We have studied all the techniques. We studied X-ray, we studied CT scan, we studied PET scan, we studied functional MRI, right? And then there were 2 more techniques. So you just need to know 5 of those techniques, and if you write one paragraph for all of those, you will get 10 marks. Okay. December 2021, this question came, elucidate ablation and histological methods to study the brain. 5 plus 5, which means for ablation you have to write for 5 marks and histological methods you have to write for 5 marks. And then December 2020, this question came, write short notes on the following, neuroimaging techniques and electrical stimulation. So we have covered every topic that can potentially be asked in the exam. Okay. So to now conclude what we have studied today, the different methods to study biopsychology. What are we doing here? We are trying to understand the invisible. What are the invisible? Your thoughts, your feelings, and your behavior. How? By making them visible. How? Through all these methods that we have studied under methods to study biopsychology. Okay. So that was all about this topic. I hope I could simplify this for you. If you have any doubts regarding this, please feel free to reach out to me on the comments. Subscribe to the channel to know when I'm going to put the next video. Let me know what you think about the video by liking it, or maybe drop me a text and share it with your friends also whom you think might need this. That's all for today. We have a pretty long video for this topic. So yeah, I won't be taking any more time. Take care, have fun, keep learning. Bye bye.